My name is Fatih Sarioglu. I'm an associate professor of electrical and computer engineering at Georgia Tech. I have always been very passionate about developing technologies for scientific measurements. Therefore, I feel truly privileged to have received an award named after Dr. Arnold Beckman. Dr. Beckman's seminal inventions that you can find in almost any laboratory today certainly inspire me as an engineer. The huge impact he made on science with these inventions motivates me to be better at what I do. In addition, his general approach to life, emphasizing integrity and humility above all, makes him more than a professional role model for me. The focus of my work, supported by the Beckman Foundation, is to develop electronic biochips for measuring the properties of cells in a biological sample. Cells are the building blocks of life. Complex organisms like humans contain many different cells that work together in harmony. However, it often takes a small amount of pathogenic or malfunctioning cells to develop infections or deadly diseases like cancer. Therefore, it's critically important that we have the tools that we can analyze cells and differentiate them not only for detecting diseases early on, but also to enable scientific research on those cells for developing effective therapies. Existing laboratory tools to analyze cells are problematic in several ways. First, they are not accessible beyond specialized laboratories. They're expensive, bulky, and require well-trained staff to operate them. Second, generally only good at measuring a single cell property. This prevents a comprehensive analysis, especially needed for complex diseases. Because two cells can be deceivingly similar if you look at one parameter, but they can be different when you start looking at more. To address this need, my students and I developed portable biochips that can quantitatively measure a variety of cell properties and present the results as digital data, which can easily be stored or transmitted via mobile phones. Our biochips are built using the same techniques traditionally used to manufacture computer chips. But instead of using expensive materials, we use plastic and glass to make our devices low-cost, disposable biomedical tests. Inside every chip we build, we create channels, as thin as a single human hair, to move cells from one place to another. As cells move in these channels, we subject them to a variety of tests. For example, if you would like to assess how stiff a cell is, we pinch the cells by narrowing the channel and measuring the time it takes for them to clear the constriction. Or if we want to find out if a specific protein is expressed on the cell membrane, we bring the cell to the part of the chip coated with an antibody with an affinity to target cell protein. If the cell is captured on that surface, we then understand that that specific cell is positive for the tested protein. To track how cells respond to these tests, we equip our biochips with an electrical sensor grid. As cells move in the chip, they flow over the electrical grid and change the electrical current between electrodes. To differentiate between signals coming from different parts of the chip, we design different electrode patterns at different places to produce distinct, location-specific signals. We then use machine learning to recognize these signal patterns in the output signal, and this way we decipher cells' responses to tests and compute their properties. I should mention that the technique we developed to follow cells on the chip is based on years of research in telecommunications, and those same ideas are still being used for routing signals in mobile phone networks. We have already used our biochips for a variety of measurements. For example, we measured the expression of tumor-specific proteins on human breast cancer cells. It was very exciting to see that the data from our small disposable biochip matched with those from a giant laboratory instrument worth hundreds of thousands of dollars. We also developed blood tests to analyze white blood cells and measure the prevalence of different immune cells. Such measurements are currently possible only in clinical laboratories and form the basis of many clinical decisions. In another study, we were able to differentiate between different cancer cells by measuring their elastic properties on our biochips. This demonstrates the potential of the technology for accessing non-traditional cell biomarkers in basic research. In summary, the Beckman Young Investigator Award allowed me to develop portable and low-cost biochips that can measure a multitude of cell properties. These devices will enable researchers to comprehensively analyze cells by measuring different cell parameters on a single platform. Such an analysis will identify cell types that cannot be differentiated by a single parameter and help develop new diagnostic and therapeutic strategies. Furthermore, the simplicity and automated nature of the technology will allow it to be utilized widely by non-specialists in technology. Finally, being low cost and portable, these biochips are extremely well suited to be utilized for diagnosing diseases at the point of care, especially in low resource settings. Dr. Beckman's inventions enabled chemical parameters to be converted into quantitative electrical data, and this revolutionized biochemistry. Similarly, my aim in this project is to convert biological data from cells into quantitative electronic measurements. As Dr. Beckman said, my hope is that eventually I can also do more for science by making these tools available to the scientific community than I can do in my laboratory by myself. I would like to conclude with thanking Arnold and Mabel Beckman Foundation for believing in me and my work. I'm happy to be part of Beckman family.